We are going to talk today about a recent paper that we got accepted for publication in the Journal of Physiology. And the first author of the paper is Shailee, who is with us today. It's the first time that we're going to actually have the privilege of having Shailee in one of the videos that we do in the lab, because Shailee has been a PhD student here for a few years now, and she's just about finishing. She's going to defend her thesis, so she has 3 very robust publications. And the last one, which is the one that we're going to talk today, is about insulin resistance or obesity-induced insulin resistance in muscle, in the skeletal muscle of females. So it's going to be a good opportunity for us to look at the female side of the complications that we have with obesity. And also, obviously, we have Daniel. Daniel has been with us before, and he's a co-author in the paper and also understands quite well everything that we did, and he has also some important insight to give us about the study. Okay, Shailee, so what was the main goal of the study, and what are the important findings that you got when you did in males and also later on in females?
So one of the key mechanisms that actually induces insulin resistance is the accumulation of lipids in muscles. So it's this intermediary lipids like DAG and ceramides, which are known to cause lipotoxicity.
Sorry, DAG is diacylglycerol, right? Okay.
So we looked at, after giving these rats a high-fat, high-sucrose diet, we looked at 3 different muscles based on their different oxidative capacity. And our hypothesis was different muscles with different oxidative capacity would have different accumulation of these intermediary lipids.
So a muscle that's more oxidative would probably accumulate less of these intermediary lipids because it would divert those lipids towards oxidation, versus a muscle that's less oxidative would probably accumulate more of those lipids and be more susceptible to the development of insulin resistance.
So oxidative capacity would be more protective, as opposed to a high glycolytic muscle would be less protected.
More susceptible to insulin.
Okay.
However, we found that all the 3 different muscle types we had developed insulin resistance, and they all had accumulations of DAG and ceramides in male muscles.
We had that observation in males. We said, okay, let's see if in female muscles that have a different amounts of fat and also different muscle mass, as males have 40% body weight is made up of muscle, females 30%. So if females have way more fat than males. So does that affect, the idea was, does that also affect the way the muscles, the oxidative and glycolytic muscles manage lipids?
And also females, in terms of skeletal muscle insulin resistance research, is not something that's been studied extensively or as extensively as in males.
There is definitely a gap in the literature, so we wanted to look at females and understand. We found that with the diet, females, all 3 types of muscles, we looked at Soleus, CDL and Epitrochlearis in females also, and we saw that they all developed insulin resistance. However, the only driving factor that caused insulin resistance in female muscles was DAG accumulation. There was no alteration in ceramide content with the diet. So this was a key
muscles,
difference.
in summary, muscles and male muscles, you had DAG and ceramides. In female muscles, was mainly diacylglycerol or DAG accumulation that drove insulin resistance. Now, we also studied another factor that is crucial for insulin resistance in obesity, which is
Inflammatory markers.
inflammation.
Yeah.
So for that, we measured.
We looked at a few inflammatory markers such as TLR4, TNF-alpha, interleukin-6. We found that inflammation was the driving factor for causing insulin resistance in glycolytic muscles for males, but in females, it was in EDL and Epitrochlearis that we saw an upregulation of inflammatory mediators.
So a mixed fiber type muscle and a glycolytic muscle. So 2 different types of muscle in females and only the glycolytic muscle in males.
Yeah.
So it is fair to say that when you study one single muscle, you may identify insulin resistance. You don't have a distinction in mechanisms because not all muscles are are affected mechanistically the same way.
Muscle is responsible for the majority of glucose disposal with insulin stimulation. And so what that means is it's a crucial tissue for the regulation of whole body glucose homeostasis. Now, from the other perspective of things, looking at both males and females, we're able to identify distinct underlying mechanisms that regulate insulin resistance in these muscles. Although the functional outcomes, the development of insulin resistance, is the same, the underlying mechanisms are different. And so the development of any therapeutic intervention should be developed accordingly, keeping in mind that certain mechanisms affect muscles in males and other mechanisms affect muscles in females.
Okay, so do you think that, is there any consideration we could make with exercise for this, from this point of view? So do you think that exercise could be something that both in males and females would be a useful therapeutic approach or something to think of?
I think there is the possibility for the recommendation of certain interventions that might effectively be used for both, including exercise and potentially even a dietary intervention, which in males and females, for instance, the ketogenic diet has been shown to improve whole body glucose homeostasis and insulin sensitivity as well.
So I think Shailee did some work in that
Yeah, so
area,
I was about
right?
to say, like, interestingly, with exercise, there is a higher accumulation of DAG, which is called the athlete's paradox. However,
Is it DAG or TAG in
both.
athletes? In
It's
both.
It's
Okay.
DAG and TAG both, which is why it's called an athlete's paradox. And we're not sure how, if DAG is supposed to induce insulin resistance in athletes, how there is a higher accumulation of DAG. So there is recent publications and recent studies that show that it is actually the species of DAGs that is inducing insulin resistance in some cases and some not. It's not the total DAG content.
Okay, so when you say, is it the same? Because I thought the species would be relevant for ceramides, but is it for DAG? Do we have different species of DAG too?
Yeah.
Okay.
So DAG and ceramides both, it's hard to make assumptions based on total ceramide or total DAG content. It's actually the specific species that may or may not induce insulin.
Okay, so when you mean species, can you give us a little bit more of explain what these species are? I mean, what would make a different species of ceramides, for instance, and a different species of DAG? Because DAG is diacylglycerol, 2 fatty acids in 1 glycerol.
So it's essentially isomers, like a different structural format of the DAG or a different structure.
So it would be a different type of fatty acid, you mean, that is esterified to the glycerol molecule.
Yeah.
So depending on the type of fatty acid, seems to be more detrimental, some others less detrimental.
Yeah, like C18 species and C16 species in both DAG and ceramides have been found to be, let's say, the more toxic kind of ceramide species or DAG species. It's just the longer the chain, I think it causes insulin resistance.
Okay, so from a dietary point of view, then, if we can avoid, because the DAG will be formed based on the fatty acids that you eat in the diet. So if you can avoid those types of fatty acids, you might be able to
Navigate.
this is a hypothesis, obviously we haven't tested it. Okay, but you did some work in muscle also looking at ketogenic diet. And apparently, if I remember correctly, when you use a high-fat, high-sugar diet, you have an increasing insulin resistance. But if you use a ketogenic diet, which has as much fat, if not more fat, but you take away the sugar, now you preserve insulin resistance. The muscle don't develop the same condition. So it looks like there's also a combination of different macronutrients that can also affect it. So it's not we usually say high-fat diet, but we should actually specifically mention high-fat, high-sugar diet or high sucrose diet, because that's the one that seems to be more detrimental to metabolic health. Thank you very much. So we'll stop here and I'll see you in the next. Bye for now.